Good morning, everyone, and I hope you all enjoy the midterm break. I think this is the first machine learning seminar after the break. And today I'm very happy to invite Associate Professor Xia Ning from the Ohio State University to our machine learning seminar. And as usual, we have 45 minutes talk, follow up with a 15 minutes question and answer. So I will hand it over to Yunxin to introduce Xia. Thanks, Yunxin.
Thanks, Kai-Chi. And thank you, Xia, to actually provide this very interesting talk today. And so a little short bio. So Dr. Ning and I had worked on, along with Kai-Chi here, worked on this last year. And Dr. Xia Ning is an associate professor from the Biomedical Informatics Department and the Computer Science and Engineering Department at the Ohio State University. So she received her Ph.D. in computer science and engineering from the University of Minnesota, Twin Cities in 2012. And her research is in artificial intelligence and machine learning with application to drug discovery, healthcare, and e-commerce. So her research is highly interdisciplinary, and her lab develops innovative and effective AI models and computational methods to derive knowledge from heterogeneous big data, conduct modeling, ranking, classification, prediction, and generation, and ultimately solve critical and real high impact problems. Specific application includes new molecule generation and drug candidate prioritization for drug discovery, cancer drug selection, and for precision medicine, and drug repurposing for Alzheimer disease, and information retrieval from electronic medical records. So Ning has also won 10 years highest impact paper award at IEEE International Conference of Data Mining in 2020. This is a very prestigious award, and congratulations on that. So without further ado, I'm going to pass it over to Xia to talk about her talk today. Thank you.
Thank you. Thank you for the introduction. Let me share my screen, and I hope you can see my screen. Let's see if we can do full screen. Okay, can you see my screen here? Okay, thank you. Thank you for having me here. So this is Xia Ning. I'm an associate professor from the Biomedical Informatics Department here at Ohio State University. And my background, as Yunxin just introduced, is on computer science. So I want to talk about our most recent work on generative models for molecule optimization, and hopefully can give you an idea how we leverage machine learning, deep learning, artificial intelligence to address some very traditional problems into a different domain. So before that, I want to introduce something called Artificial Intelligence Index published by Stanford University in 2020. So this is the index in 2020, and this year there is another new version for 2022. I think this is very interesting because it lists 10 different kind of trends about AI. And the first one is in AI investment in drug design, the discovery increased significantly. So this is the trend in the US, but I feel this is probably the trend in the entire world. Like people in the last few years, people invest a lot on drug discovery, like for cancers and for many other diseases using AI. This is definitely a very strong trend. And in the third item here, it's about generative everything. Like previously, people use generative models to generate images, to generate audios, videos. Now, actually, it's everything. And our work is actually on the first and the third item here. We want to do drug discovery using AI, and we use generative models. And some other also very interesting trend about AI. So this is a very brief outline of my talk, and just to give you a quick introduction about drug discovery process. This is not a very typical computer science thing, but you will see, hopefully, there are many interesting problems that really require some computational approaches and advanced AI to facilitate the entire process. So the typical drug discovery starts from the identification of therapeutic targets. Like we have a disease that we want to study, and the first thing is we need to identify which disease is related to this, which genes are related to this disease. And after that, we want to identify some potential drug candidates. So oftentimes, typically people do very high throughput screening just to quickly test among, for example, 1,000 drugs, whether there are molecules which can show potential efficacy against the genes. And among those promising potential drug candidates, people do some optimization, meaning that they want to change probably the molecules have some efficacy, but not good enough, or the molecules have some other toxicities. So in this step, people do the optimization and they change the molecule structures a little bit so as to improve the desired properties and hopefully remove those undesired properties. So this is about the design and the synthesis. And after that, hopefully we have 10, 20 different small molecules, and those molecules can go through the cell culture assays to test their efficacy in cell lines. And a few of them will go through the animal testing and get tested on those mouse, small animals. And hopefully we have 1 or 2 drugs which can go through the clinical trials. So typically, we will have 3 phases in the clinical trials. The first phase is about safety or toxicity. So to make sure that the drug is safe among the participants. And then in the second phase, it's about efficacy, or whether the drug are effective in treating the disease. And in the end, it's going to be about efficacy and safety again among probably a slightly larger population or participant groups. So hopefully after these 3 phases, we can have 1 single drug that is successful and send it to FDA until FDA approved. So this is a very typical drug discovery process, and typically it's going to be about more than 10 years and costs probably more than $1 billion now. But as you may know that this process has a very high risk. Like oftentimes we don't even have 1 single drug out of the entire process after a decade. Recently, there is the new drug repositioning paradigm to accelerate this drug discovery process. And the idea is we want to use some FDA-approved drugs to treat a new disease. For example, now people are working on identifying some drugs to treat COVID, some FDA-approved drug to treat COVID. So the key here is to identify some signals which can tell us whether this drug can be used to treat a new disease or not. So it's about the identify the new therapeutic indications of existing drugs. If we can find such signals, for example, from electronic medical records or from clinical trials or from scientific publications, then that drug can go through quickly go through the clinical trials only in phase 2 and phase 3, because for phase 1 we don't need to test it again because it's FDA-approved drugs, it's already safe. So we just need to test the efficacy of that drug for the new disease. And after that, hopefully that drug can be FDA-approved for the new clinical use. So this is the drug repositioning paradigm, and the good thing about it is it's much faster and less costly. But the key thing here is to identify the new therapeutic indications. So in my lab, actually, we do a lot on the entire drug discovery pipeline, both the traditional pipeline and the new paradigm. So for drug screening, we have work on molecule property prediction and virtual screening, and molecule optimization and synthesis. So I'm going to talk about optimization today, but we also have work on identifying synthesis path of a given molecule. And on cell lines, we have cell line response prediction. And on clinical trials, we constructed something called clinical trial knowledge graph, so that you can leverage all the methods over knowledge graph in order to do inference or in order to discover new knowledge out of the graph. And for FDA approved drugs, and we do post-market drug pharmacosurveillance and we do the drug adverse event prediction identification. So particularly if someone is taking more than 2 drugs. So in our data set, we have people taking more than 10 drugs. So if multiple drugs together, oftentimes their side effects are not really observed in clinical trials. So we need to have ways to detect and also to predict drugs, to predict the side effects for multiple drugs. And also for drug repositioning, we have work on drug repositioning for Alzheimer's disease, and we try to identify the, again, signals for repositionability from electronic medical records and from drug-gene interaction networks. And overall, we have worked on electronic health record data analysis and information retrieval. As you can tell, in this entire drug discovery pipeline, there are many interesting problems, and probably advanced AI methods can be developed to address these problems. For example, how to identify drug candidate. So the entire chemical space is about 10 to the power of 60, that is the size. But among all the molecules, all the small molecules, we want to identify just 1 or 2 particular molecules for a particular disease. That's not a trivial task, actually. And for any given molecules, we want to predict their properties using, for example, their molecule structures. And this is also a very interesting problem, and also very recently, like in deep learning over graphs, they're having increasingly more amount of work on molecule property predictions, like we want to predict the bioactivities or toxicities or other other properties that molecule have for them to be a successful drug. And selecting different drugs for different patients, this is probably the primary goal for precision medicine. And drug selection is also something really interesting. So it requires some ranking or prioritization techniques to identify probably the one particular drug that is best for each patient. And adverse drug effect prediction, as I just mentioned earlier, and the therapeutic indication prediction to identify whether a drug can be used to treat a different disease. And in silico molecule optimization and the generation, this is particularly for molecule optimization in the very early phase of drug discovery process, and giving a molecule how we can synthesize the molecule. So this is about retrosynthesis prediction. So I'm going to talk about the optimization problem today. So before that, let's look at an example here. So if you're given these 2 molecule structures and I ask you to change the left molecule structure to the right one, probably, I mean, by looking at these 2 molecular graphs, we can tell, oh, let's just remove these 2 chlorine atoms and include a fluorine atom. So it's probably pretty easy for us. But the question is, what happens if we need to have computers to do the job, and the computers have to do this type of modification automatically? Probably the first question is how these molecule structures can be represented, and in a way such that our AI method, our machine learning method can operate over. So of course, there will require some representation learning over small molecule structures, molecular graph structures. And the next question is, what happens if we need to modify millions of molecules, not just 1 or 2 of them, but millions of them? So there must be some high throughput type of modification algorithms that can be done, and all the representations of molecules and pattern recognition of those molecules should be done automatically. And also, probably the next question is, what happens if you want to modify this molecule into another molecule such that this new molecule is very similar to this original molecule, but with some difference, and the new molecule has better properties. So if we're looking at toxicity, the new molecule should be less toxic than the original molecule. So how can we do that? This is about, some motivations behind our generative methods. So again, some background about molecule optimization. So this is a kind of early step in the drug discovery process. Like, we have some promising drug candidates, but not good enough. So we want to change their structures to make them better, to have better properties of this molecule. And often, conventionally, it is done through something called fragment-based screening or synthesis. Like, we just collect all the molecules that look similar to this molecule drug candidate, and test all of them and see if we can find a better one, or we synthesize a new molecule. But recently, people try to use deep learning to facilitate this process. Like, we want to use deep learning to do everything. Like, for if you must have heard the AlphaGo, that is to use AI to simulate the protein structures. Now we want to use AI to modify molecule structures, and people are using like some different ways to represent molecules. So molecule graphs is probably the most straightforward way, but there is another representation called small strings. So if you remember, each chemical structure can be represented using a kind of sequence of those atoms or sequence of the symbols. So we do have the string representation for small molecules, and probably, as you can imagine, people are using natural language processing methods to deal with small strings. And also, there are a lot of generative models. And for example, people can connect atoms and bonds in order to generate a new structure. And these are very popular. Actually, starting from 2018, there have been a lot of work on using AI, particularly graph neural networks, to address many problems in drug design and drug discovery. And some issues with this deep learning method, like many of them cannot return the desired scaffold or desired major structures of a molecule. They can generate a totally new molecule. That's also useful, but the issue is this is not going to be the thing that people will use in real drug discovery. They start from something promising, and as in the drug fragment-based screening or based method, they change the structures of something of the kind of starting point, starting molecules, rather than identify totally new molecule. So that means in our generation method, we need to make sure that the desired scaffold, desired substructures or fragment of the molecules should remain. And also, many methods just optimize the molecule structures with respect to one or two properties, and we want to do more than that. So that's the kind of, again, motivation of our method. So let's see, again, before I show you the method, here are several keywords of some key ideas of our generative method called Modof. It's a one-fragment based generative model. So when we modify a molecule or change the molecule structure, the first thing we want to do is we want to predict something called a site of disconnection at a molecule, and only modify the molecule by changing the fragment at that site. Or in other words, giving a molecular graph, we just want to change a small portion of this molecule graph. And the first step is to predict which portion or which bond of this molecule that we want to change from. And in order to learn all the disconnection site and also learn how to change a molecule, we need to learn, we need to learn from the training data, right? So in our case, our training data, each training instance consists of actually 2 molecules. They are similar, except they are different. They are very similar, but only different at one particular bond or at the site of this connection. So we learn from the difference between the molecules, and we consider one molecule is optimized after the other one. So we learn the difference from the molecules before and after the optimization at just one disconnection site. And actually we encode the difference into the latent space. And when we generate, when we modify the new structure, when we modify a molecule, we do the sampling from the latent space and generate one fragment that can represent the expected difference. So this is about the generation. And given that, what we can do is we just remove the fragment at the disconnection site and attach the generated fragment at that site. So it's very intuitive, and actually it's very similar to what's really happening in the drug discovery in the one fragment based modification. Since we have the latent space, we can do multiple sampling, and that can give us multiple optimized candidates. So you don't want just one possible candidate, possible solutions. You always want to have more. So this is to generate more optimized candidates. And as I just mentioned, our method can only optimize molecule at one disconnection site. But if we combine our model into a pipeline, each module in the pipeline optimize one fragment, but the entire pipeline can optimize multiple fragments at the molecule. And we further optimize our pipeline such that at each module we can generate more candidates. So the entire pipeline can generate many different optimized molecules. So giving all the background, all the intuition, all the key ideas, here is the formal definition of our problem. So given a molecule MX, we want to optimize this molecule into another molecule MY, but with under these 2 constraints. The first constraint is the new molecule, the optimized molecule should be similar to the given molecule. And there is a similarity constraint saying that the similarity should above a certain threshold. Or in other words, in terms of their structures, they should be similar. The majority of their structures should be similar. And another constraint is the optimized molecule should have better properties. So if we have way to measure properties like P of and Y, the property of the new molecule should be better than the property of the old molecule. And this is going to be very general for all the given properties. So this is a overall architecture of our generative model. So actually, this is an autoencoder type of framework. So we have the encoder part and also the decoder part, and also we have this latent space. As I just mentioned, the training data, each training instance is a pair of 2 molecules. So in this particular example, these are the 2 molecules. They are very similar and only different at one particular atom. So in this example, they are different here. So in the first molecule, the carbon is connected to a nitrogen here, and in the second molecule, it's connected to a ring. So this is the only difference between these 2 molecules in terms of their structures. And in terms of the properties, the second molecule is always better than the first molecule in a particular given property. And we represent molecule structures using their graphs, molecular graphs. So each atom is a node and each bond is an edge. So naturally, this is a small graph. We also construct another representation called junction tree. So in graphs, we have rings, and many algorithms, over graphs do not like rings, right? So, but in a tree, we don't have rings. So it's easier for us to run some algorithms. So we generate something called a junction tree, but this is more or less a dual representation of the graphs. So each bond in the graph, for example, this blue bond in the graph becomes a node in the tree. And if 2 two bonds are connected through a common atom, then the corresponding nodes will be connected in the junction tree through a patch. Some special thing about ring. So each ring in the molecular graph becomes a node, and if some bonds are connected to this node, then in the junction tree, the corresponding node will be connected. And also something special about this carbonate here, because it's connected to other 3 carbonate. So it's it has 3 bonds. So if an atom has 3 bonds, then that atom becomes a certain node in the junction tree. And if all the bonds, I mean, it's going to be connected to all the other nodes in the junction tree, which represents the bonds. So this is how we can convert a graph into a tree structure. So these are the training data. And during training, we learn a difference, and we encode the difference embed the difference into a latent space. And in the decoder part, given a molecule that we want to optimize, we sample. We first do a sampling from the latent space and use that latent vector, latent embedding, to modify this molecule step by step until the final result. So there are multiple predictions involved in the entire process, and so during the training, it's going to be end-to-end. So it's, the loss function includes all the prediction loss, and encoder decoder is a typical autoencoder training process. And in the testing phase, we just need to use a decoder. Like given a molecule that we want to optimize, we use the decoder. And again, as I mentioned, sampling from the latent space to decode or to optimize this molecule until the final result. So this is a kind of overall architecture of our model. So let's take a look at the encoder. So the idea of this encoder is to learn the difference of 2 molecules which are only different at one particular bond or particular atom. That is actually our site of disconnection. So the training instance, each training instance has 2 molecules. The first thing is we need to represent the molecule structures in order to learn something from it. We use graph message passing neural networks to represent graphs. Actually, for each node in the graph, we do the message passing to aggregate the information from its neighboring nodes and to generate an embedding of that node. So this is learned from graph message passing neural network. And actually, similarly, in the junction tree, we also do the tree message passing neural networks. It's more or less the same message passing thing, but in this case, we learn the embeddings of each node in the tree. Actually, that corresponds to each bond in the original graph. And after we have all the representations of all the nodes, then we can compare the difference of these 2 junction trees. So in this particular example, actually, these 2 junction trees are only different at here. Like in the first junction tree, it has a purple node connected to this dark blue node, and in the second one, it has the light blue and the light green nodes connected to the blue node. So the difference of these 2 molecules are actually is actually only here. So that's why we use the node embedding of this dark blue node and this purple node in the first junction tree to represent its difference from the second one. And in the second junction tree, we use the embedding of this dark blue node, this light green node, and also the light green node and the light blue node to represent the second junction tree. And there actually, these 2 vectors will represent the difference among these 2 molecules. And we map these 2 vectors into a Gaussian distributed, into a latent space following Gaussian distribution. And this is, you can consider this latent space as a space to encode the difference of the molecules. The first half encodes the different different portion of the first molecule, and the second half encodes the difference of the second molecule. So that's the encoding part. So in the decoding part, giving a molecule, we represent the molecule using its junction tree structure, and we want to modify this molecule into another one. So the first step we do is to predict a disconnection site, or to predict where the modification should be happening. So for each of the node in the junction tree, we do the prediction, and we pick the node that has the highest probability as the disconnection site. So in order to predict the disconnection site, we use the embedding of the node. So in this case, if we want to predict for the green node, the dark blue node, we use its embedding learned from the junction tree structure, and then we do a sampling from the latent space. So we use these three vectors together to do the prediction. So intuitively, you can think whether this node is a disconnection site is a function or is related to its neighboring structures, which is encoded in its embedding. And also, quote unquote, it depends on a particular rule that change the molecules based on Z minus and then Z plus, or remove something from the molecule based on Z minus and add something based on Z plus. So we use these three embeddings to do the prediction. And let's see if our prediction says this dark blue node should be the disconnection site. Then the next question is which part of this blue node that we should remove. So the next prediction is the remove fragment prediction. So this node is connected to an orange node here and a purple node here. So we need to predict whether this orange node part should be removed or this green node should be removed. So for these two nodes, we will have predictions and predicted scores. So we use the embedding of the, if we look at the purple node, we use the embedding of the purple node and the Z minus to predict if we need to remove that or not. So again, the idea is we need to look at the local structure around that purple node, and also this Z minus tells us kind of rule, quote unquote rule, or whether this should be removed or not. So we use these two embeddings. Let's say our prediction says we need to remove the purple node. Then after that, we can represent our result. I mean, actually, after the node is removed, the intermediate result in this way. So actually, this is our intermediate representation, but they are not really valid molecules. We still need to change this structure by adding some things back to the structure. So starting from these intermediate representations, we first predict whether we need to predict some other structures that can be attached to this structure and modify this structure. The first thing is we want to predict whether we should add something or not. So it's likely that up to here, it's a good molecule, we should stop there. So the first thing is we want to predict whether we should stop or not. So this is based on the junction tree structure of our intermediate results. And if our prediction says we should stop, then we stop. But if it says we should add some new structures, then the next prediction is what kind of structures, the structure type. So here, the structure type is predicted again using the information at this particular node and also the information from the entire junction tree structure. And let's say we predict a particular atom type that should be added at this particular attachment point. Then the next question is where to attach. So in many cases, it's pretty straightforward. You just connect it to a particular node in the junction tree, which corresponds to an atom in the graph. But if this tree represents, if this node represents a ring, then among the multiple atoms in the ring, there could be multiple different attachment points. So only in that case, we need to predict where the new fragment should be attached to. So we do these predictions multiple times until our prediction says we should stop. This is the final product, the final result. And of course, we always do some valence checking and make sure that it's feasible to add a particular node. It's for example a double bond or single bond or triple bond at a particular atom attachment point. So that's the decoding part to kind of modify a given structure into another structure using the latent space, latent embeddings, and also the structures of any intermediate results. So here is the data set that we used to train our model. So actually, in our model, we use about 55 or 56K pairs. Compared to the other baseline method, actually, this is not a huge number. Actually, we don't really require as many training data points as other methods because our model is actually less complex compared to other methods. But this table gives you some quick idea about data sets that we use. And the table 2 shows some overall comparison between our method and some other baseline methods. So among the baseline methods, these 6 different baseline methods, probably as you can imagine, they use, there are kind of 3 different categories. The first one is to do the VE type of encoding, decoding. It's autoencoder type of framework, but encode the entire molecule structures, and the decoder part will decode a new molecule structure. So the issue with that type of framework is the decoded molecule can be totally different from the input molecule. And there are other kind of reinforcement-based method, like I believe GCPN and Graph AF. They are based on reinforcement learning. So the modification is formulated as a way to add atoms and add new fragment, and of course you will have the reward, that is the desired, that is the properties of the final product. So that's the reward in the reinforcement learning, and you can train the policy network. And MALFlow is another type of method. It's based on the flow framework. Like in autoencoder, actually you encode something into the latent space and decode that, but the encoding and decoding part are not really reversible. But in the flow-based method, it's invertible mapping. Like if you map something into latent space, you can map that thing back from the latent space back to the input. So that mapping is invertible. Our method is different in terms of training, in terms of the architecture, because we train from pairs, and this method always train from single molecules. So we compare our method under different similarity constraints, like delta zero. Delta as 0 means we don't really care about the similarities. But if we use a very high similarity threshold, that means the output, optimized molecules should be very similar to the input. So overall, we can see if we don't have any similarity constraint, these numbers are the properties. So our proper improvement can be really huge. And if we require a lot of similarities, like we constrain that the results have to be very similar. In this case, our method can still achieve a much better property improvement compared to the baselines. So in this particular example, the properties is called P log P, which measures the hydrophobicity of molecules and also the size of the molecules, and also the things, kind of rough measurement of synthesizability. So that means if we constrain that the optimized molecules are very similar to the input molecule, our optimized molecules can have better properties in terms of hydrophobicity and complexity. So this is probably the major conclusion out of our experiments. And I want to show you some very typical fragment that we remove and we attach in our experiments. So we learn our model in a 100% data-driven way. We don't really have any chemical rules or chemical expertise integrated in the learning process, but this is what we learn from data, and actually we present this method, these results to our chemist collaborator, and they told us it makes sense because like for example, including some aromatic rings can always improve the property like hydrophobicity or that type of properties, so they make sense. So this is more or less a validation, independent validation after for our results. And also want to give you a quick idea about how the molecules are optimized step by step. So let's say this is the first molecule that we want to optimize. In the first step, the small fragment is attached, actually it's replaced at this point, at this ring location, and that increase our property from -5 to -2.6. And then we add another aromatic ring at this location, at this point, and that increase the properties again. And actually, in the next step, we add another ring with 2 small bonds here, and the properties further increased. And then a small fragment is added at this ring, and properties further improved. And in the last step, it's another small chunk, small fragment, small bond at a different location here. So this is more or less a step-by-step modification through our method. And as I mentioned, each molecule, our method only modifies the molecule at a particular location, at one fragment. But if we combine them into pipelines, we can do multiple fragment modification. So here is another example of multiple disconnection site, multiple fragment optimization. So this is the molecule to start with. And in the first step, we somehow this weird structure is changed to a ring structure, and that change can dramatically increase the property from -3 to 0.4. And then in the next step, another small fragment is added at this aromatic ring, and that increased the properties a little bit. And then in the next step, actually, instead of changing on the left-hand side, the method moves to, the modification becomes on the kind of right-hand side. So there is another small fragment is added here, and that increases the properties a little bit more. And then another fragment here, and then in the last step, actually, it goes back to the left-hand side of this molecule, and another small fragment is added on the left-hand side. So this is just to demonstrate that we can use multiple identical modules of our method to modify multiple fragments of a molecule. So probably those are the major conclusions and major results. And in terms of conclusions, our method modifies one fragment at a time. So this is probably more similar or approximates what's happening in the in vitro chemical modification, because people only modify one fragment at a time. And by doing this, actually, we can our method can better inform the in vitro molecule optimization, can tell people what to do next by doing the prediction. And our method only encodes and decodes the fragment that needs modification, and we don't really encode the entire molecule structure, so we only encode the difference. So you can interpret, or you can think that the encoded latent space represents quote-unquote rules about how to change molecules. And so we can do multiple fragment modification using a pipeline, and actually by using this pipeline, we can control we can control some and really understand or interpret some intermediate modification step, and also can give us some interpretation as to see how the molecule is optimized step by step in the end. And our model is actually less complex than the state of the art, and it's about 40% fewer parameters and 30% less training data. And in terms of the performance, actually, we can achieve much better performance compared to the other baseline methods, particularly when the similarity constraint is really strict, and we can still get about 9% improvement. And if we leverage multiple sampling strategies, we can even have better performance. And we can optimize molecules with respect to different properties, like with respect to a given target. Like in this case, this is a particular protein target, and then we can optimize the molecule structures and properties with respect to that particular target structure. And we can also improve the drug likeliness of the molecules to make the molecule more drug-like. So it's multiple optimizing properties that we can optimize with respect to. And I believe these are the major conclusions and some references for your information. And we published our source code. And also, if you're interested, I do suggest that you can take a look at our supplementary materials. It has much more than what we have in the main manuscript. Like we discussed how to identify those pairs for model training, and how to what is the best way to measure molecule similarities and their effect on the molecule generation process. And also we identify some issues among the existing work, like sometimes people report their results, but it's on a different testing set, on a much easier testing set. And as you can imagine, their performance is better if you just look at the numbers, but they are on a different test set. And also for some reinforcement learning method, we also identify some issues which actually make their performance better than they should be. So a lot of discussions from there. So if you're interested, I do suggest that you take a look at the supplementary materials and also try out our data, our code. So that, I believe that's all about the generative model, but I would like to take this opportunity and do some advertisement, because we are looking for students. And so PhD students, postdocs, and visiting scholars, and the students in in my lab are all from computer science department. It's a PhD students in the computer science department, but we can also recruit students into Biomedical Informatics Department or the College of Public Health. And in my lab, we also do many other things, like we do a lot on recommender systems, like how to do sequential recommendation, basket recommendation, and use knowledge graph-based method, and also attention-based method. So we're also looking for people who to do the research on recommender systems, and molecule optimization and retrosynthesis prediction using AI, using probably as I discussed today, and also, as you can imagine, using a lot of reinforcement learning method for retrosynthesis and for peptide optimization. So peptide is a small amino acid sequence. So we want to generate new peptide. So it's a lot of reinforcement learning method. And also we have ongoing projects on in general AI in healthcare, like electronic medical record analysis, and some mobile health applications for Alzheimer's disease. So we're looking for people on all these three major areas as PhD students or postdocs. So the postdoc positions are available immediately. And we're always welcoming visiting scholars. So I believe that concludes my presentation. And if you have any questions, I would like to take them now.
Yeah, thank you, Xia, for the exciting talk. So let us first ask if there are any questions from the audience. I do have some questions. Sorry. So basically, I'm not sure how to measure the property constraints and how those constraints are considered in the generative model?
Right. So for all the molecules, actually, that's a good question. So for all the properties, we have kind of standalone predictors to calculate or to calculate the properties. For example, if it's the let's see, where is it? So if it's log p, then we have another algorithm to calculate the log p property. It's not part of our model, but it's a standalone model. And if it's drug likeliness, we have ways to calculate drug likeliness. And those properties are kind of leveraged in the optimization process. Like the loss function is about the predictions, like the the autoencoder, whether we can decode exactly the molecule that we want. So that's part of the loss. And also there are a lot of predictions, like whether we should stop or not, it's a prediction. What kind of atom types should be added, that's a prediction. So in the loss function, it's all about the loss, the losses from the predictions. Once we train the so when we train the model, we always make sure that once we add something, the property is improved. So it's kind of also our objective, but not in the objective function. And when we do the decoding, we always watch for the property and see if the property is improved. If the property is not improving, then we just stop there. So that's the idea. That's how we elaborate, how we utilize the property in our method.
Right. Thank you for the answer. I think, yeah, that's what I want to ask. So another question is, when we generate the fragments, how do we know we should stop or not?
Yeah, that prediction.
Yeah.
Yes, that's prediction is also learned, right? So we have a function to predict in the current substructure, given the current structure at a particular atom, whether you should add something or not. Right. I should have mentioned that during the training we use the so in each prediction, when we train each predictor, we always use the ground truth. Like after we add something, we use that to train our prediction. It's a teacher's forcing kind of strategy, right? So the training, we always use the ground truth. After we add something, if our predictor says we add A, but the ground truth is B, there is a loss for that particular predictor. But after that, we still use B, the ground truth, for the next prediction. So that's the training part. But in the testing phase, if we want to decode a molecule, then it's always based on the predictors. So as you can imagine, an issue could be if we made a mistake earlier, then it's going to be wrong in all the following steps, right? But that's something, we don't really have the reinforcement learning type of facility in the decoder part, so that's a kind of sequential decision-making thing. But during the training, we do have the ground truth and the teacher forcing.
Yeah, it makes sense to me. If you mention, teacher forcing, I immediately get the idea. So so there is a question from the chat. Does the method also learn a task order optimization task? Among all the bounds, which bounds should be updated first to reduce the total number of subsequent replacements?
Yeah, we don't really learn the order explicitly. So the first thing in the decoding phase, the first thing we need to do is to decide where to modify, where to start from, and from there, yeah, I think I know what you mean here. So after we add a particular node, for that particular node, sometimes in general, it can be it can there could be multiple node attached to that particular node. So that's it's like after we add a node, the predictor says we need to add this node, we add that particular node, and then for that node we predict we should stop here or not. If it says we should add some more nodes, then we add some more node. At that point, after we add additional node on that particular node, we still need to predict whether we should stop there or not. So if the predictor says we should not, that means we need to add another node to this particular node. And then the predictor says, oh, we should stop here. But remember, we add another two particular node to this particular node. So for these two other new node, we still need to predict we should add something or not. So it's like, if you think about it, this is similar to the kind of breadth-first search in the graph, right? So you add until you need to stop at this particular node, then you move to all the added node and to the similar thing. So in that case, I mean, literally we don't really explicitly learn the ordering, but implicitly there is an ordering. It's a kind of following the breadth-first search ordering of the graph. I hope that makes sense.
I've got, can I ask a question? Yeah.
Yeah, sure.
I've got two questions. One is a little bit more specific, and is generally from an interest point of view, because a lot of machine learning researchers will always have the similarity constraints, like the delta you have on this page. And depending on different algorithms, they set this very differently. And I see that in your results, you had 3 or 4 values for this that you in your experiments. Is there a specific way that you typically would set this for molecule optimization? Or is it a little bit more general in terms of this is an exploratory based technique? So we just have to explore different delta values there.
For molecules, sort of, we have some ideas about, I mean, the thresholds like 0.6. I mean, for molecules, actually, that's not a very low threshold. That's not a 0.6 means the molecules are already very different. Like 0.9, it's going to be super similar. So we don't really have a way to say which threshold is good enough, but we sort of know which threshold means how similar they are. Like, so still, I mean, we cannot really, I mean, we still try different threshold and see the performance, but how to set threshold, which threshold to use, it's, I think, up to the users.
Thank you. The other question I have is what I realized, and you sort of alluded to this as well. One of the things that typically is very hard because we're using all this deep learning stuff is the interpretability of it. But it seems that your technique is interpretable. Would you classify your technique as interpretable?
Interpretability. So it really depends on how we define interpretability, right? So here, when we say it's interpretable, we mean we know exactly what happens in the entire modification process step by step, right? We know at this step, this is the atom, this atom is added, and the next step is this bond. We know exactly what's going on in the entire optimization process. From that perspective, I would say it's interpretable. And also in the pipeline, we know this module optimize this fragment, and next module optimize this fragment, and next module is optimize the fragment. So that is interpretable. So from this perspective, I would say we do have some interpretability. But from a different perspective, like why this atom is attached in this way, we don't have that interpretability. We don't really, I mean, it's the predictor itself is still a black box, right? It gave us this prediction, but we don't really know why it's predicted in this way. So from that perspective, I would say we still need some interpretability, particularly related to chemistry or chemical rules. There must be some chemical rules. It's a so I think in the end, if we can map that to the chemical rules, that's going to be really great. But as for now, it's still a black box.
Thank you so much.
Yeah. So any other questions from the attendees? No. If no, I think we can wrap it up. And yeah, and thank you again, Xia, for presenting the exciting research to us. And basically, I think many of the students and researchers in our departments, they are quite interested in using in developing, designing graph algorithms. I'm also doing something on graph neural networks and also doing some deep generative models. For example, using deep generative models for trajectory generation.
I see.
So there's slightly different, but we also considered to use graph neural networks on some spatial data, GPS data. So I think the techniques behind are quite similar. And I'm very interested in the talk and yeah, so very interested in any further, discussions and collaborations.
Yep, definitely. So so in our case, it's small molecules, but in the end, it's graphs, right?
Right.
So it's very similar techniques, although it has to be customized for different applications.
Right.
Yeah.
Right. Yeah. So do you think you have anything to add?
No, I just want to thank you, Xia, again. I appreciate her taking her time to actually come and talk to us. Actually, this will be uploaded on the web somewhere.
Right. I think after the talk, the recording will be uploaded to, I think it's YouTube, and we have a link from our homepage to the YouTube video, I think.
I see.
Yeah.
Cool. And I think a lot of other students that can't make it today would be interested in watching.
I guess it's because of the morning tea.
I don't know what you're
We have a morning tea on Friday morning.
Oh, I see.
Yeah. It's our first week back to university at the moment.
Okay, I see. It's okay.
Cool. Thank you again.
Yeah.
Thank you for having me.
Yeah. Thank you.
Yeah, we would love for you to come visit us one day.
Oh yeah, I would love to do that in person.
Right. Okay. So thank you very much and see you guys.
Okay, bye.
Yeah, thank you.